pressure, or even electric fields, for example. That is also an issue that, for example, in fish is very pronounced. But today we're going to focus on chemicals, and that is taste and smell. And if we think about ourselves, then it is quite obvious that we have different organs that are basically responsible of taste and smell. So we have noses, we have tongues, and the question then is, what do other animals do in order to perceive taste and smell? And this is something we are going to explore today. So perceiving these modalities in generally, that requires receptors. And these receptors that can either be very specialized and respond to only a single modality, or they can be also multimodal or polymodal. So they integrate more than one modality. And then it gets a little tricky, as you will see. Just an example from our human life is that free nerve endings, for example, they respond to temperature or to mechanical stimuli, so pressure, touch, strain, and in general, we will call those pain receptors. But when we think about an animal, and here I just pasted in some Agiope brunichi, as you will see, female and male. There is a lot of chemical information in a very complex world. This graph is taken from Fleischer, and it's, as you might notice, it's more meant for, let's say, insects than spiders. But the sources where this chemical information comes from is very similar for all types of animals. So if, for example, it can come from competitors, for example, other species, these informations, they can come from conspecifics, for example, mating partners, and that is something that we, I think, are very interested in. So how do mating partners interact? What kind of information can a mating partner perceive from a potential partner? But other topics like colony members, for example, for spiders that are colonial or that are even social, that is also a very interesting topic to pursue. Other sources are more also basic sources of chemical information, that is how to acquire food, how to find, this is certainly not something that pertains to spiders, that they need to find plants, but they need to find animals that they can feed on. But they could also, let's say, feed on carrion. That is also something that could be interesting, and maybe this is more prominent than we think of. Yes, or they find conspecifics as food and so forth. Then oviposition sites, that is, I think, a field that has been little explored as to spiders, because the question is, how do they find a good place where they lay their eggs? It's not always in the web itself, because it can also be outside. So how is that decided on, where the eggs are oviposited, and how they're provided with shade, with shelter and so forth. And a very important point is how to perceive predators, or can they perceive predators? Can they avoid predators? And how is that? How do these dynamics actually work? And there we do not really know a lot if we think about it. So spiders do have chemical sense. We know that they are able to taste and smell. That is something we know because we have some information on all these topics that are listed here. But in general, the chemical sense is important for detecting prey, luring prey, detecting avoiding predators, recognizing conspecifics, detecting and choosing mates, choosing the habitat, all what we just saw in that graph that I presented. The chemical sense, I think, is especially interesting, or I would like to start with, actually, with whether and how they can recognize conspecifics and then detecting and choosing mates. And how can they do it? They don't have noses and they don't have tongues, but they have lots and lots of sensilla that are of different shape and length. And this is just a model of sensilla on these spiders. And the question then eventually is, which sensilla are responsible for the chemical sense? I would like to mainly focus on our pet animal here, on Argiope brunichi, because we have so many data on it. And so do not be surprised that I will talk a lot about Argiope brunichi. I will here and there also mix some other results, some other studies in between. So what do we know on Argiope brunichi? We know a lot about its behavior. That's why I think it's interesting to focus on this species. The males are attracted to females from a distance. We know that. We know that males can distinguish conspecific females. We know that female odor triggers male maturation. We know that odor reveals relative female quality. And we know that odor reveals relative female age. Or maybe absolute. We don't exactly know that. And this is a very recent finding. It was published, or it's a preprint by the group of Jutta Schneider, who can be seen here sitting in the field observing Argiope. And she found with her group that odor or better taste might actually reveal relatedness, because the cuticular carbohydrates that are found on the body surface of the Argiope, they differ depending on relatedness. So that is at least a possibility that they also can discriminate between relatives when it comes to mating. So how are these results assessed, or how do we gain these kind of results? It's observations in the field, obviously, but also choice tests in all sorts of arenas. So one such classic choice test, that is a choice test where males are released on a bamboo stick, for example, some kind of a stick that is connected to silk threads from the female webs. And then the male walks along that stick, and then he gets in touch with both silken threads from these two females. And then hopefully what we see is a decision that the male makes. It either goes to the right or to the left-hand side, and certainly one has to control for side effects. But here you can see a male blander that is depicted here, that is in a situation where he can choose between a female blander and a female Argentata Argiope. And what happens if we have several rounds? I think in this case we had about 20 trials, and you can see here in percentage that these males, they choose the conspecific females. So that's actually a clear-cut signal, a behavioral signal, that they can discriminate between these conspecific females and the non-conspecific females. Okay, but what does it tell us? We don't really know if taste or smell or both are involved, because when the male walks along here, we can not really disentangle whether it's smell, olfaction, that actually attracts him to one or the other female, or whether it is only taste, meaning taste of some information that he gets from the thread when touching it. Another question that you might ask, looking at these the slide here, is that the question could be, is the presence of the female required? Yeah, is it the web only, or is it the female only, or is it both that are releasing an information for the male? And another question could be, is it actually the silk texture that makes a difference? Maybe it's not at all anything related to chemical information. It could just be that the threads of the conspecific female are different from the non-conspecific one, and so the male just decides on the texture alone. Or maybe it's the diameter, but that could also be. So I'd like to to tell you a bit about this topic by introducing another study that is tetragrammaton. And that's a study I did together with Ashley Adams from Berkeley and other people. And what we did here was a very simple experiment where Tetragnatha versicolor from California and Tetragnatha extensa from Central Europe, from around Greifswald, were tested against each other. And we used male Tetragnatha extensa as the males that were asked to decide on where to go. So what we did here, we removed the females. So the choice trial is actually a choice trial that is only related to spider silk and not related to female presence. But what we also did was, and you can see that the conspecific ones are very strongly preferred over the heterospecific females. But what we also did, and you will see that in the next slide, is that in order to avoid this texture or diameter related topic, we took extracts of the web of females and then used cotton threads that certainly were of similar size because it was one and the same kind of cotton wool. And these cotton threads were then impregnated with the extract of the web. So we could exclude the possibility that something that's related haptically to the choice is excluded. But we had the same effect, so this is a very clear-cut result. The extract showed that the males would prefer the conspecifics over the heterospecific females. Now, how do they differ, these silks? These silks differ slightly, but significantly. For here, you can see a representation of the gas chromatograph of an extensa and a versicolor female. And you can see that there are, it doesn't really matter what it is in detail, but that there are compounds A, B, C, and D, and they are distributed non-equally over these profiles here. So extensa has A, B, C, and versicolor has D and B. So there is a clear difference in these gas chromatographic profiles here. And these profiles, or what we see here, these peaks, they relate to methyl esters that need to be investigated as to whether it's really them that are causing a differential response of the males. But that is certainly not so easy because you would need to produce a synthetic compound. First of all, you would need to produce the potential pheromone synthetically, and then having that, it requires a bioassay where you actually test whether the synthetic compound is really responsible for the reaction that you see. Okay, so and this is not trivial at all. So the knowledge on these active compounds and really tested compounds is very limited. But as I mentioned before, we have plenty of behavioral evidence that there is something out there, that there are a lot of volatile and contact pheromones that are important for spiders and for probably all spiders that are out there. So what is known? This is a relatively old, I have to say, compilation, and it's not complete. As you can see, it's from 2012. But just to give you an impression that there is not much out there, because it hasn't really changed that much over the time, over these 10 years. There are a few species, Agelenopsis and then Pholcus and Argiope, whose actual substances that are biologically active are known and that they have and that have been studied in the field, and also with these synthetic compounds. So that is actually a small selection of evidence that we have here, and then others were investigated and considered to have contact contact pheromones that are possibly volatile. It's not always easy to distinguish between the two. But as you will see here from this table, there are not that many either. So if we compare this to other animal groups, then the knowledge we have on the Arachnida is really deplorable. So 1% of pheromone studies where the actual pheromone was identified relates to the spiders or to arachnids in general, and certainly we know much more on lepidopterans. We know a lot on beetles, and we know some on some hymenopteran groups and some mammals, obviously, and Diptera. So this is maybe not surprising because these other groups are more relevant for us humans when it comes to pest species. So there is more money invested into knowing how to lure them, how to maybe kind of take them from our agricultural fields and so on. If we look at what we know in spiders again, this is also super tiny here when it comes to all the different groups here, but never mind. What is important here is that you can get an idea of how diverse these structural compounds are, these pheromonal compounds, and you can see that they are very different when we compare species. Copienus is a bit similar to Argiope, as you will see from here. It's also a citrate compound, but apart from that, you can also see the diversity is enormous, and there is little possibility to link these different structures, and it's also very unclear how they evolved in the course of the evolution of spiders. Okay, let's focus on Argiope again. We have investigated the chemical profiles of different life history stages, that is subadult, virgin, and mated females of Argiope brunichi. And we find that these chemical profiles of the silk extracts, of the body extracts, and of the headspace analysis are different between these different life stages or reproductive stages. And that can be, as I mentioned before, relatively easily done with gas chromatography and mass spectrometry. And in our case here, the question is, what is the difference? So headspace analysis certainly is something that is a good idea to do, because you can actually catch all the volatile substances that an animal produces. And you don't have this confounding effect when you're producing extracts of silk or body, that it could be that there are some volatiles in there, but it also could be that you have contact compounds that are broken up and then kind of seem to be potential volatile substances. Anyways, headspace analysis, that is, as I mentioned, a good idea. And what we see here is that virgin females, they really have this one super high peak of a substance that mated females don't have, or at least in very, very low number, and subadult females also don't have. So this is a very nice result. That's something one would wish, yeah, and often it's not the case that it's so simple, because we know from many insects that these chemical profiles can be very complicated. And it could also be that a whole bouquet of different compounds is important for attracting a mate, for example. But here it seems relatively simple. So what is it? This peak A, it's this structure here, and it's methyl trimethyl citrate. And so here comes a little complication, because there is stereochemistry involved. And this is also noted from insect, that sometimes it is important in what ratio these enantiomers are actually occurring in the pheromonal product that is produced and sent out into the air. So here, the natural ratio between these two enantiomers that I simply marked in blue or red is 6:1, and up to 25:1. So that is a relatively flexible natural ratio. That might mean that it's also not so important how exact the ratio is in terms of its attractiveness to the male. What we did then is, and that is a complicated process, to synthesize this this compound, and the synthetic compound was then tested in the field in a similar but also lower ratio than we found in a natural setting. And that didn't matter so much, and that is nice that it didn't matter so much. Otherwise, it would be very complicated. So what we did was that we put out the synthetic citrate in the field, and we had so-called tripod traps. So we had these little skewers, wooden skewers, and they were covered in Vaseline, and that's because the Vaseline keeps the compound in the solvent a bit longer. Otherwise, it would just evaporate very quickly. So what we found was that when we put the citrate on such a trap, it attracted males relatively reliably. And we had always, together with a citrate trap, we had a control trap that was only provided with a solvent. And in those traps, they had no effect. There was no males attracted. So that was also a very clear-cut result. So clear-cut, something like I haven't really experienced many times during my scientific career, that there were simply no males interested in this solvent control traps. On the other hand, when males arrived at the citrate trap, they actually courted that trap, so as if a female was around. So they really made courtship movements on that parafilm and a paraffin cover, and also around the trap on the leaves nearby, like this male does at the moment. So we also then looked at the question of how little material is needed, and we then went down in the amount that we applied to the traps more and more. We diluted the amount to less than 1 microgram. And here you can see the number of trials we performed and how successful they was, and how many males we attracted. So you have to consider that this was out in the field, and so it all depends on whether there were males around, yeah, because we had to go to different places every time we put up a trap. But it shows us definitely that that the citrate functions as a volatile pheromone. We tested it because of the synthetic compound was active, and it was even active in very low amounts. But then how do they do it? How is olfaction and gustation, or let's say chemical sensing in general, possible? How do they do it, these spiders? Which structures are involved? And now I like to present you the situation as it is in insects. In insects, this has been investigated much more than in spiders. And the typical the typical syncyllum that is used for gustation, it's certainly also all about syncylla. The typical syncyllum used for gustation is a so-called tip port syncyllum. So it's a syncyllum that only has an opening at its tip. That's why it's called tip port. On the other hand, for olfaction in insects, the typical thing to use is a syncyllum that has wall pores along the shaft. And here you can see a cross-section in that area here. Sorry, my pointer is a bit slow. And you can see that there are openings. So any kind of order molecule that is biologically active can enter here at almost any stage or any piece of shaft here along that syncyllum. So that is something that is typical for insects. And now let's look at spiders. What kind of receptor functions do they have? What kind of structures do they have? They certainly have slit syncylla. They cannot be the ones that are responsible for chemosensing. They have trichobothria. We know that they are airflow detectors. They have tarsal organs, and they have been suspected to be also involved in chemosensing, but that I think is now off the table. They have a lot of tactile hairs that are touch receptors. And then the question is, amongst that mass of syncylla, which of those are the chemical receptors? Yeah, so, oops, there is something. Let me see. Okay. Something, okay. It's a little slow. Okay, so we have actually some evidence that wall-pore syncylla do occur in some arachnids. This is known from some amblypygids and also from some acari. And here you can see syncylla that really have some openings, some pores, and inside you will find the dendrites of these neurons here that are chemosensory active. But this is an exception, or let's say at least for the spiders, this is not to be found. There are always exceptions, but the general situation is that there are only tip pore syncylla. From what we know, in the Arañae, there is only tip pore syncylla as candidates for chemoreceptors. Now, what do they look like? They certainly look similar to the tip pore syncylla in insects. There is a shaft that is typical of every syncyllum. There is a tip pore that you can see here. Here in Argiope, there is shaft ridges, so they can be a bit diverse depending on the species that you're looking at. They often have this S-shaped curvature here that you can see here, and certainly they have a very pronounced socket. So if we look through the legs, for example, of Argiope brunichi, and we try to find out if there are differences depending on where we look at along the body or along the legs of the spiders. This was done by Anne Saraganske in my group a few years ago, and she found only tip pore syncylla in Argiope brunichi. So you can see here a few tip pore syncylla. They all look a little different here because there is such a variety. Sometimes these outer structures are not so easily visible, but it's just one type. It's just tip pore syncylla so far. Maybe, I mean, you never know what we will discover, but it looks as if there were only tip pore syncylla. Now, this led people that are used to this distinction between tip pore syncylla are for gustation and wall pore syncylla are for olfaction. It led people think that spiders can only taste. You will find that in some literature once in a while that this is the suspicion, but as we have plenty of information on the behavior of spiders, we know that they can. We just don't know exactly how they do it. So what we set out to investigate is a very, very general scheme of spider chemical sensing. And this is a current project that is funded by the German Science Foundation, and that we started in a team of people that are also present today. That is Carsten Müller, who is the specialist in ultrastructure in my group. So he's looking together with Bilal on the anatomy of syncylla. We're looking at the ultrastructure of syncylla on different body parts of spiders, and we're also including a comparative aspect here. And the behavior, I mentioned a bit about the behavior. The behavior is now a bit outsourced or is running in parallel at the University of Hamburg in Jutta Schneider's group. She is interested in or she took over the topic of pheromone release dynamics, on whether pheromone is stage or age dependent, condition dependent. And as I mentioned before, she also published a preprint on relatedness. So in our group, so this is our focus here, the anatomy of the cincilla. Then we are looking at the electrophysiology and the pheromone receptors, but I will come to that later on. I just wanted to show you a few faces here of the people that are involved and also partly present today. And if we now go to the topic of gustation, yeah, we can say, okay, everything that a spider should touch is touching during his life, yeah, that should actually be done with areas that are capable of gustation. So maybe we can turn the whole story around and say, okay, what is actually in touch with the substrate? And if parts of the body are never in touch with the substrate, but have tip pore cincilla, we can probably assume that these are structures that are capable of olfaction. Maybe that's too simple, but we will find out. I think it's a good start and a good guess for the time being. So this is something Bilal also looks into. What I mean is that he is looking at the behavior also of spiders during walking, during foraging, during mating, and then checking what parts of the legs are in contact with the substrate and which parts are never in contact with the substrate. But that is something that I will not show you more about today. Yes, so what we would expect is that there should be gustatory receptors on the tip of the pedipalps. That is what spiders are using to touch the ground, to touch to touch prey, to touch potential mating partners, and also tips of the legs and also mouth parts are possibly unlikely involved in gustation. Behavior suggests that this really is the case, because when we look at spiders and they are, let's say, they are intercepting or they are fed with animals that are not palatable or repulsive, that they also show this behavior. Yeah, they throw out the bug that is not really pleasant to digest for them. So they also seem to discriminate between new and old carcasses. They discriminate distasteful prey, as I mentioned before. So, but it could be that there is more fine-tuned discrimination possible. We expect that the possibilities of discrimination are much more diverse. And there is certainly some evidence from electrophysiology, but not much, that there is a discrimination between salt and sugar and amino acids and so forth, but not that much has been done on the matter in spiders. What is necessary before one starts exploring the diversity of these cincilla is to know where they occur. And that is something that Anne-Sarah Ganske did in my group a few years ago. So we know exactly, at least for Archaeope brunichi, where the tip pore cincilla occur. So everything that is blue here, I hope you see these tiny dots, these are all chemosensory cincilla, meaning that these are all tip pore cincilla on these podomeres of the spider. So knowing where they occur can also then help us to investigate what do they look like at the tip of a leg, what do they look like in an area that never gets in touch with the substrate, and so forth. And is there a difference between the different legs and the pedipalps? Is there a difference between males and females? But now let's have a look at some SEM pictures on the cincilla. Here, every little star here shows that there is a tip pore syncyllum, and I already showed you what they look like. So we can explore this very nicely with scanning electron microscopy, but then also proceed to transmission electron microscopy. And this is, as I mentioned, what Carsten is an expert in. And here we have the part of the results of a paper that we published last year. And here you see that when you cut through such a tip pore syncyllum of Agape brunichi, it actually looks very similar to what we find, what we know already from Arañas diadematus. So that is a good result, that it's at least within the Arañiids, it's very similar. And what is very diverse is the number of receptor cells. So there can be about 20 chemoreceptive cells, but also some mechanoreceptive cells. To show you such a tip in a TEM section, that was a super happy moment for Carsten when he cut through this tip of that tip pore. You can actually see the dendrites as they go up, and you can see that there are several dendrites in there. So that is quite obvious. So these are the chemoreceptive dendrites, and if we look at a schematic drawing that Carsten did, you can see that here is the syncyllum drawn, and you can see the socket again, the shaft cuticle, and the tip pore. And the chemoreceptor cells are now given in yellow, and their number can be very varied, as I mentioned here, from 2 to 23 were found. So this is one thing. This is nice because we know, okay, this should all be dendrites that are somehow chemically receptive, but we still certainly don't know whether they are involved into olfaction or gustation. But what is a bit difficult when it comes to spiders, is that they also have mechanoreceptor cells, as I mentioned before, and they are here. So this is the orange part that is highlighted here. And that means that whenever we are trying to do electrophysiology from these syncylla, we are likely also getting a response from these mechanoreceptor cells. So there are two functions here. One is chemoreception and one is mechanoreception, and that makes our life pretty difficult. And that there is mechanoreception is proven here by the tubular bodies that we find here in the socket region. Okay, so we have two sensory modalities in one, so that's one thing. And what we're going to investigate here is whether these syncylla that look very similar from the outside are actually different in ultrastructure in the inside. So if on different podomeres there is a variety or a differentiation in kind of cryptic anatomy, if that's allowed to say. And we are then also going forward into a comparative aspect. So let's now talk a bit about the electrophysiology. And the electrophysiology is, as I already insinuated, quite tricky. So we want to test whether there's a neural response to a pheromone, and we expect that there could be a disparity in function depending on where on the body we're actually looking at these syncylla. So we are trying to combine the internal anatomy and the electrophysiology, and then also add some pheromone receptor analysis to it. So what does that look like? Electrophysiology, what you could do, for example, is to get some potentials from let's say a pedipulp. And in insects, this would normally be an electroantennogram, an EAG, but in spiders that do not have antenna, we call it an EPG, an electropalpogram. But this means that you have this some potential and you cannot really distinguish where is the action coming from. So it's kind of a combined response that you actually get when you're measuring that. And that is okay for the start. And what we found in Arañas diadematus, that is a paper that was just published in Nature of Science, that we found that female Arañas diadematus, they actually react to pheromone puffs of other females. So this is something I think that should be considered and should be explored further, that not only males are reacting to females in that they are attracted to these females and maybe also can base their mate choice criteria on that pheromone amount or whatever quality it can really reveal, but that also female can potentially react to other females since they seem to react to the pheromone themselves. Electrophysiologically. It could be that there is some female-female competition that originates from that or is connected to that, that females might move sides depending on how much competition there is, or they might increase the signaling when there are other females that are signaling, or they may mute themselves. This is something completely unexplored. And I think that's quite exciting to know. Okay. Yes. So this is the synthetic pheromone test that we did, not just air puffs, but synthetic pheromone tests in Argyre peperonici are possible because we have the pheromone, we know what it is, and we can apply it in such a setting. So we have made some reaction tests this summer, and they are currently being analyzed, and so I cannot really tell much about it, what the results were there. But the other possibility of doing electrophysiological work on this system is that we can try to do single cell recording. So as mentioned before, the sensilla might be different in internal structure, but also in how they react to different cues or signals, depending on where they are. So maybe there's a disparity in response. So how does one do that? So one picks out the sensilla that one knows to be tip-pore sensilla, and then you can put an electrode here at the base of that sensillum and see if it reacts to a puff or to a substance that you apply. Keep in mind that because they are mechanoreceptive, you might get a lot of signal from the mechanoreceptor, but not so much from the chemoreceptor. So that has to be disentangled. Okay. Yes, so these are the electrodes, obviously, and you need an amplifier and you need a screen, and then you certainly also need a puff. And if you're lucky, you get a response. So this is something that has been done already, and that this almost 20 years ago. And the only electrophysiological study on a spider that was a single sensillum recording was done on Cupienus by Harald Tichy in those days. And you can see here that the sensilla look quite different from the Argiope ones because they are pretty hairy, but they also have a tip pore here at the tip, a pore at the tip. So how did he do that? He had a measuring electrode, obviously, and a grounding electrode, and an amplifier and a screen. But what he did was that he had a contact stimulus. So he brought the contact stimulus or a stimulus in contact with the tip of the pore, and the stimulus was a so-called cupulure. That's also a citrate compound that was gathered from the female silk and was also then synthesized in the group of Stefan Schulz in those days. But I mean, it was successful. That is super, because apparently they did not have a lot of interference of the mechanoreceptors, but we don't really know exactly whether this citrate is really a contact stimulus or also an olfactory stimulus, because it was only tested in contact. So there are some things that will probably need to be revisited. So the approach is to look at the disparity in function, as I mentioned, the disparity in ultrastructure, but another possibility is also there, and you might have thought about it yourself already. Maybe they are not so different. Maybe a single syncyllum can be used for taste and olfaction. So that is certainly also a possibility, that within a single syncyllum, we can have olfactory sensory neurons and gustatory sensory neurons, and that is all bundled up in these several neurons that we find within a syncyllum. But let us then move on to the chemosensory receptors, and that is something maybe that can be revealing in the end. And what do I mean here by chemosensory receptors? So we have chemosensory neurons, and within these neurons there are receptors, and these receptors are within the membrane. They are proteins, and so within these proteins are then kind of capturing the odorants, and there's a signal transduction, there's a nerve firing resulting from that, and some perception going on, and maybe a behavioral response. So what do we know from insects again? And we always have to go back to insects because there's so much more known than in spiders. So there are several superfamilies of proteins, and they have some confusing names because these are considered to be olfactory proteins, unless although they are termed here, for example, gustatory receptors, olfactory receptors, and ionotropic receptors. And this is relatively well known for insects by now, but certainly the details are still explored in many different laboratories at the moment. So what did we find? So we did a transcriptome analysis of Agape brunichi and Agape blanda, of male and female pedipalps and legs, and we found no homologs of insect chemoreceptors whatsoever. So that was a bit worrying. But it seems that chelicerates have their own repertoires, and this is also a result of a paper that I cite here, that the chelicerates are kind of different from the insects when it comes to their chemoreceptors. There are olfactory receptors. This is gustatory receptors, olfactory receptors. There are potential homologs of some specific receptors that are known from insects. But if one blasts them manually, then one can find so-called kainate receptors. And this is now for us kind of a window into the transcriptome receptor protein analysis, that we are trying to tackle these specific gustatory receptors that seem to be chelicerate specific, and these olfactory receptors that seem to be from the potential homologs of ionotropic receptors or kainate receptors. So what can one do with this? How does one proceed? One can do an in situ hybridization and a functional assay, which is on the way at the moment. And in parallel, we are also working on the transcriptome of all the legs, because before we only had a selection of legs. So we're going through all the legs, and we're going through the pedipalps and everything separately, and we're looking at what is actually expressed in which leg, and to what degree, and is there a difference between these podomeres, or these legs, I have to say. So what can one expect? In insects, this is relatively well established. You can have an in situ hybridization protocol if it works out. What you see here, for example, in this moth, is that you can actually see the light in the tunnel, and you see that there is an expression of a certain olfactory receptor here in the antenna of this moth. And this is certainly something that I'm dreaming of to produce for spiders, so that we know which receptors are actually expressed where in the spider, and then we would hopefully see whether there is this disparity in expression over the course of the legs, or when you compare different legs with each other, and also between males and females, and so forth. So there is a lot of potential here. Another interesting approach is an in vitro expression system. I think I might take 2 more minutes. An in vitro expression system, and that is done, surprisingly enough, maybe, in a frog system that is Xenopus oocytes. These Xenopus eggs, they are very convenient since they're very robust. And what you do then is that you inject RNA of a given receptor, and this receptor is then produced within the oocyte of that or within that frog egg, and inserted into the cell membrane. And if that works out, and you're not ruining the egg, but that this process can actually happen, then after some days you can test if the oocyte is responsive to the odorants that you are interested in. So that frog egg responds to spider odorants in the ideal situation. And you can then explore that with a 2-electrode voltage clamp method. So this is what I wanted to show you today. I touched upon all these different aspects that are important for spider chemical sensing. The behavior, the anatomy, the electrophysiology, the pheromone receptor topic, and what we are hoping to be able to develop a bit more is not only that we know how do they do it, these spiders, but also how did the chemosensory system evolve within spiders as a taxon. So what is the origin and the evolution of the chemosensory genes? So that would certainly be a next step when comparing what there is within spiders with, let's say, for example, insects, crustaceans, and so forth. That will certainly not be my expertise, but there are many people out there, and especially people that we work together with in Lund. The most challenging questions, I find. So who is involved here? This is Christa Löfstedt from the pheromone group, the leader of the pheromone group in Lund in Sweden, and then the people that are doing the electrophysiology and the transcriptomic analysis and oocyte recording, that is Dan and Hong Lei. And from my lab in Greifswald, I already mentioned Carsten Müller and Bilal. And Carolina will be involved in the transcriptomic analysis. So thank you very much for your attention. I'm sorry if it took a bit longer than expected, but that's how it is. I'm looking forward to your questions.